Manuel, I think we will continue with the second lecture and we will have time for discussion at the end, right?
Yeah, yes, absolutely. We will move to the second speaker. Please remember all of you that you can be sending questions to the speakers through the Q&R button throughout all the presentation, and we will have time to discuss them later. So now it's my absolute pleasure to present an excellent scientist and a good friend of the laboratory, Professor Ruben Nogueiras. He completed his doctoral thesis in 2003 at the University of Santiago de Compostela here in Spain, under the supervision of someone that you all know, Professor Carlos Dieguez. Subsequently, he undertook a postdoctoral training in Germany, Switzerland, and US under the supervision of Matthias Schopp. Since 2009, he coordinates the molecular metabolism group in the Institute of Research of Santiago de Compostela. In 2014, he obtained a position of associate professor. More recently, in 2020, he has been a research professor opportunius at the University of Santiago de Compostela. From the scientific point of view, his group is focused on the study of the molecular mechanism involved in obesity and its associated comorbidities, including type 2 diabetes and muscle D. That is the topic that is bringing us all here today. More particularly, he and his laboratory investigate new pathways involved in both glucose and lipid metabolism in the liver and their association with the progression of liver diseases. With a basic background, they are also determined to figure out which is the translational implication of their investigations using preclinical studies and clinical data. He has co-authored more than 290 publications in international journals. He has received a lot of national and international awards, and he is principal investigator of many different projects at national and European level, including some ERC grants. So with this CV, you can imagine that we have received a great lecture about interaction between metabolic pathways and muscle D. So if you are ready, Ruben, it's a pleasure for us to have you here.
Thank you very much, Manuel, for the very nice introduction. It's very clear that we are friends, so appreciate it. Thank you very much to the European Society of Endocrinology for the invitation to be here, and welcome everyone, and thanks for staying there in this afternoon. So what I'm going to talk about is about some metabolic pathways that are important for the development of muscle D. So I'm so happy that Elisabetta already showed this slide, very similar one, showing the progression of the disease, starting with steatosis. Then a percentage of patients can develop inflammation, NASH, then fibrosis, cirrhosis, and finally hepatocarcinoma. Of course, not all patients with fibrosis, cirrhosis, or hepatocellular carcinoma have steatosis in the beginning, and of course not all people with steatosis will develop fibrosis or further stages of the disease. But pretty much this is the scheme, and as Elisabetta also highlighted, it is the most prevalent liver disease in our days. It affects to 30% of the general population. So the questions are, which are the parameters responsible for this disease? And in the beginning, there was a theory which was called the two-hit hypothesis. And it was quite simple. it said that the accumulation of fatty acids in the liver caused liver damage, and that was responsible of the second hit, which was inflammation. And this inflammation was the main responsible of steatohepatitis and all the further liver damage. So this hypothesis is partially correct, but it oversimplifies the real situation. And nowadays, it is widely accepted the multi-hit hypothesis. So we now accept that there are many different factors involved in the progression of this disease, and these factors are very heterogeneous. So, for instance, a very important factor is the diet, and Western diet is normally very rich in glucose, fructose, and fat. And it is very well known that the ingestion of glucose will produce insulin resistance, not only in muscle, as it is represented here, because muscle is the main organ responsible for glucose uptake, but also in other tissues like the adipose tissue. Also, fructose is doing a detrimental action in the liver because it promotes de novo lipogenesis, and obviously this synthesis of new fatty acids is going to lead to the accumulation of fatty acids in the liver, which is the first hit. And fat intake, again, is going to cause the increase of this amount of lipid content in the liver. Then there is a strong interaction between the liver and other organs. A very obvious one is the adipose tissue, because when the capacity of storage of fatty acids within the adipocyte is overwhelmed, this excess of fatty acids will go to other organs in the body. One of those organs is the liver, and therefore the lipid metabolism in adipocytes, it's very important. It plays a very relevant role for the detrimental action in the liver. Also, the secretion of some factors like leptin, adiponectin, different adipokines can affect the amount of lipids in the liver. And of course, the inflammatory status of the adipocyte, especially in obesity, is going to produce also an effect in the liver. Another important organ for the liver is the intestine. First, because the gut microbiota is now very well known to play a key role, because metabolites derived from the gut microbiota can be released to the circulation, can reach basically all the organs in the body. One of them will be the liver, and some of those metabolites derived from the gut microbiota may have a detrimental action in the liver. Also, the permeability of the intestine is very relevant, because normally if there is an inflammation in the intestine, the permeability is altered, and this is going to affect directly to the liver as well. And we have also genetic factors and epigenetic factors. So some of the most relevant targets associated with the genetic component of muscle D are the ones represented here. I will not go into detail, but all of them are somehow related with fatty acid metabolism. So mutations in any of these genes will cause alterations in fatty acid metabolism, causing the storage of these lipids in the liver. And concerning epigenetic factors, there are many, of course. Environmental factors, that includes chemicals, contamination, diet, the stress. There are many factors that normally we cannot control, and somehow they are going to induce an effect by alterations in the DNA, also by modifying the amount of microRNAs. There are several microRNAs that play an important role in the liver. All of that is going to affect the condition, the status of the liver. So the general picture is really complex. There are so many factors, very heterogeneous, and the way to study the disease is, of course, very difficult, and there are many options. One of those options, at least the one that we have chosen, was to study some genes that are important for cell cycle signaling, because it is well known that these genes are normally playing an important role in metabolism as well. And actually, some of these cell cycle regulators play a very wide role in metabolism, meaning that they can modulate glucose metabolism, fatty acid metabolism, mitochondrial dynamics, and so on. And these cell cycle regulators are in many diseases, or in many cases, involved in metabolic diseases like obesity, diabetes, and so on. So when we talk about cell cycle regulators, many of us are probably thinking in p53, because it is the most studied gene in terms of cell cycle regulation and cancer. It is responsible of many different types of cancers. And p53 is not alone. It is a member of a family of 3 members. It's p53, p63, and p73. And there are many studies showing that these 3 members can play different actions in carbohydrate metabolism, cellular redox, fatty acid metabolism, mitochondrial, autophagy, and so on. So many different factors affecting the metabolic function of the cell. And we have studied this family, mainly p53 and p63, during the last years in our laboratory. And we have seen that in physiological conditions, so in conditions that has nothing to do with cancer, these p53 and p63 members are very important for the regulation of glucose. So for instance, p53 can stimulate the production of glucose in the liver by activating one enzyme that is very important. It's called PCK1. We will talk about it later on, which is an enzyme critical for gluconeogenesis. On the other side, p63 is going to produce the opposite. It's going to inhibit the production of glucose, and that is because p63 represses the activity of sirtuin-1, which is an enzyme that is also important for gluconeogenesis. But apart from glucose, we have also seen that p53 and p63 are important for fatty acid metabolism in the liver. And some years ago, Begoña has shown that when she inhibited p53, the levels of p63 were higher, and under those conditions, the liver displayed higher inflammation, more ER express, and that was inducing lipid storage, therefore steatosis. Contrary, when she overexpressed p53, the levels of p63 were very low, and therefore she was able to reduce inflammation, ER express, and the amount of fatty acids. So if you ask, well, why these guys are studying p53 or p63, I mean, they will never become real targets to treat the disease. And I agree with that. They will not be targets to treat the disease, but what we thought is that we could learn using them as a tool, because these genes are highly promiscuous transcription factors. So they modulate the activity of hundreds of genes. So we thought that maybe using more specifically p63, we could identify new targets that were involved in the development of muscle D. And this is what we do. What we did some years ago. We did some proteomics in the liver of animals overexpressing p63, just in the liver. And in those conditions, we found some proteins that were upregulated, others were downregulated. But one of the proteins that was upregulated was ATG3, autophagy-related gene 3. As the name indicates, it is a gene involved in autophagy. And when we perform proteomics in animals where we knock down p63, the levels of ATG3 were lower. So more p63, more ATG3, less p63, less ATG3. So what we did next was to check whether this ATG3 was really important in patients. And in collaboration with some clinical teams in Spain, they provided us with liver biopsies, and we measured the expression of ATG3 in patients without liver steatosis, patients with steatosis, and patients with NASH or MASH. And we found that when we analyzed the expression of ATG3, it was increased in the liver of people with NAFLD or NASH. And the expression of the gene was correlated with the steatosis grade and the NASH score. So apparently ATG3 was maybe relevant at clinical level. And what we did was to perform different preclinical studies, and I will show just this one, where we downregulate the expression of ATG3 in animals that were fed with a choline-deficient plus high-fat diet. This means that these animals are gaining weight because they eat high-fat diet, but they have liver damage because that diet is deficient for choline. And it is very well reported that the deficiency of choline causes increases the amount of lipids in the liver. So in control mice, we found indeed that the amount of lipids was extremely high, as represented here by the oil red staining. And when we knocked down ATG3 in the liver, the amount of lipids was lower. And this was associated with increased levels of CPT1 alpha. And CPT1 alpha is an enzyme that is critical for fatty acid oxidation. So it internalizes the fatty acids into the mitochondria, and those fatty acids will be oxidized. And it is very well known that in hepatocytes, when you increase fatty acid oxidation, you reduce the amount of lipids. So in this work, what Natalia, Marcos, and Eva demonstrated is that p63 increases the levels of ATG3. This induces steatosis. And in this steatotic condition, if we deplete ATG3, we are going to activate a pathway that finally will lead to the higher expression and activity of CPT1. Therefore, we increase the mitochondrial function, and that is going to reduce the amount of lipids in the liver. In addition to ATG3, another protein that we found when we analyzed proteomics in the liver of these animals where we manipulated p63 was the mitochondrial antiviral signaling protein, MAPS. As the name indicates, MAPS is a protein that is activated or participates in the immune system after a viral infection. But surprisingly, we found that when we overexpress p63 in the liver, the expression of this protein was increased. When we measured the expression of MAPS in the liver of animals where we downregulate p63, the expression of MAPS was lower. So we did the same as in the previous case. In collaboration with a clinical team, we measured the expression, the gene expression and protein levels of MAPS in the liver of people without muscle D or people with muscle D. And the expression of this gene and protein was significantly upregulated in people with muscle D. And it was positively correlated with the NAS score and serum triglycerides. So again, we did many different preclinical studies, and I will just focus on this one, demonstrating that when we delete or we knock down MAPs in hepatocytes of mice fed with a Western diet, again, another diet that is inducing obesity and liver fibrosis, we knock down the gene, and the strategy was effective using a biogenetic approach. I will not get into that, but the strategy was good, it was efficient. So when MAPS was deleted in hepatocytes, the circulating levels of transaminases were lower, and the expression of collagens was also reduced, indicating that the inhibition of MAPS could ameliorate the damage in the liver. So in this work, Eva and Natalia showed that MAPS was inducing inflammation and lipid content in the liver. If we have a condition where the liver is already damaged and we inhibit MAPS, we can activate a pathway, and more specifically, we can inhibit an inflammatory pathway involving ERK, TNF-alpha, and NF-kappa beta. So we reduce inflammation, and therefore we improve the status of the liver. But when we check the picture of the progression of muscle D, there is one step that is very important, and it is the generation of fibrosis. And this is important because when a patient has fibrosis, it is very difficult to reverse the disease, and the symptoms are much more dangerous than when they were just steatotic. And for the generation of fibrosis, and Yelizaveta already mentioned a little bit this, there are other cells that are very important at this stage, and these are hepatic stellate cells. These are the main fibrogenic cells in the liver. So these cells are located in the space of ESE. It is a space located between liver sinusoidal endothelial cells and hepatocytes. So hepatic stellate cells are there in the middle of these two cellular types. And this means that hepatic stellate cells can respond to whatever happened in endothelial cells or in hepatocytes. Normally, hepatic stellate cells are quiescent, are inactive. But upon liver injury, they are activated. And when this happens, they transform into myofibroblasts. These myofibroblasts can produce material of the extracellular matrix, like collagens. And the production of these collagens is the reason of the stiffness of the liver. So this is the reason of liver fibrosis, the amount of collagens and other molecules of the extracellular matrix that is produced by these activated hepatic stellate cells. When these cells are activated and they transform into myofibroblasts, they proliferate, they acquire contractility, so they change the properties. So in our laboratory, what we hypothesized together with other teams is that if these cells are normally quiescent and they get activated, for this activation, they will require a high amount of nutrients to cope with the energy requirements. So it seems quite logical to think that these cells will use high amounts of glucose, fatty acids, and amino acids. And there are different articles showing that this is the case. So for instance, the synthesis of fatty acids has been reported to be important for the activation of the cells. These cells are highly glycolytic as well. So there are some literature over there. Since we know that P63 was very important in hepatocytes for modulating both glucose metabolism and fatty acid metabolism, we hypothesized that maybe P63, which is widely expressed in basically all the cells of the body, maybe this transcription factor could play an important role in hepatic stellate cells. So this is what we tested. First, we isolated hepatic stellate cells from different animal models that had fibrosis, and we used different challenges. This is a diet deficient in methionine and choline, so two amino acids that when they are not in the diet, they produce a huge liver fibrosis. These are mice fed with the choline deficient plus high-fat diet that I mentioned before. And this is a treatment with carbon tetrachloride, which is a chemical compound producing also liver fibrosis very strongly. In all cases, the expression of P63 was upregulated when these animals developed liver fibrosis. When we use human cells, the situation was very similar. So when we got primary human hepatic stellate cells from a human donor, and we activated these cells with TGF-beta, which is the classical activator of these cells, and we corroborated this, measuring the expression of fibrotic markers, collagen, SACTA2, which are the ones responsible for fibrosis primarily. So in those conditions, when cells are activated, the expression of P63 was elevated. And we reproduce the same using a cell line, human cell line of hepatic stellate cells, where we activated them with TGF-beta. The expression of the fibrotic markers is upregulated as usual, and the expression of P63 was also very high. So we generated an animal model lacking P63 only in hepatic stellate cells using one promoter, that is the lecithin retinol acyltransferase, which is a gene involved in the synthesis of vitamin A, because hepatic stellate cells normally store vitamin A. So by using this genetic strategy, we were able to deplete P63 in hepatic stellate cells. And when we fed these animals with a high-fat diet plus the deficiency in choline for 20 weeks, normally these animals are going to develop fibrosis. And this is what we show here with the Sirius red staining, that is basically staining collagens. In the conditional knockout mice, the amount of collagens was significantly lower, and we reproduced the data by measuring by PCR different fibrotic markers. Now, if we do the opposite, if we overexpress P63 in the liver, we are going to have more circulating transaminases, and we will have more collagens and more fatty acids in the liver. How is P63 doing this? Well, P63, what we found by proteomics is that was modulating many proteins involved in metabolism, particularly in fatty acid metabolism. And this caught our attention because, as I mentioned before, fatty acids are very important for the activation of these cells. And acetyl-CoA carboxylase is an enzyme that is critical for the synthesis of fatty acids. And this enzyme was overexpressed when the cells were activated. So what we did was to ectopically overexpress P63 in hepatic stellate cells. And in these conditions, we were able to activate the cells indeed, as we show here by measuring the expression of fibrotic markers. And in these conditions, the expression and activity of HCC was elevated. Now, what we did was to silence ACC in cells where we overexpressed p63. When we did that, of course, the expression of ACC and the activity was very low, and this ameliorated the effect, the fibrotic effect of p63, meaning that p63 required the activation of ACC, required lipogenesis de novo to activate the cells. How relevant is all this pathway in humans? Well, in collaboration with Javier Crespo and Paula Iruzbieta in Santander in Spain, we measure the staining of p63 in hepatic stellate cells of patients at different stages of the disease. So these are patients without fibrosis, fibrosis stage 1, stage 2, 3, and 4. And as you can see here, the colocalization of p63 was higher in hepatic stellate cells when the progression of the disease was at more advanced stages. Basically, the staining of p63 was positively correlated with fibrosis stage, NAS score, and circulating levels of transaminases. So what we proposed in this study, what Marcos and Eva did, was to show that p63, elevated levels of p63, were activating hepatic stellate cells, and this was inducing the activity of acetyl-CoA carboxylase, therefore inducing de novo lipogenesis and liver fibrosis. And this was mediated by p53 and ER2, but I will not get into details. So de novo lipogenesis is important, but how about fatty acid oxidation? Well, it is very well known that CPT1, carnitine palmitoyl transferase 1, is an enzyme very important for the oxidation of lipids because, as I mentioned before, this enzyme is responsible for the translocation of fatty acids into the mitochondria, and when fatty acids get into the mitochondria, they will be oxidized. So what we found in different animal models of fibrosis is that the expression of CPT1 was increased. And the same happened in primary human cells that were activated with TGF-beta, which, of course, the expression of fibrotic markers was elevated. The levels of CPT1 were also very high. How is this happening? Because when we treat hepatic stellate cells with TGF-beta, here in red, both mitochondrial respiration and glycolysis are elevated. However, if we inhibit CPT1, and we did that with etomoxil, which is a compound that has been reported many years ago to be an inhibitor of CPT1, we were able to reverse the mitochondrial respiration, but not really glycolysis, meaning that somehow CPT1 apparently is important for regulating mitochondrial respiration in these cells. So by doing this, we were able to ameliorate the effects of TGF-beta on the expression of fibrotic markers. If we do a gain of function experiment, so if we overexpress CPT1 in hepatic stellate cells, we are able to increase mitochondrial respiration, and therefore we are able to increase the expression of fibrotic markers. In vivo, what we did was to generate different animal models, so conditional knockouts for CPT1 in hepatic stellate cells, and challenge them to different treatments or diets. Carbon tetrachloride, diet deficient in methionine and choline, or the CD plus high-fat diet. In all these conditions, as you can see here in the right, the levels of collagens were lower than in the control animals under the different treatments or diets. How about humans? Well, in humans, the situation was very similar to preclinical models, and when we did the staining of CPT1 together with hepatic stellate cells, we found that the expression or the staining of CPT1 was elevated when the patients were at a higher stage of the disease. And CPT1 was correlated with fibrosis score and NAS score. So in this study, Marcos found that fatty acid oxidation was very important for the activation of the cells. And these cells were doing this because in this way they got more ATP, more energy. By getting more ATP and more energy, they could activate and synthesize more collagens and so on. So I will stop here and will just now thank the fantastic team that was doing all these experiments. Some of them were mentioned throughout the talk, and all the collaborators. Without them, it would be impossible to do all these experiments, and the funding bodies that are helping us to give the money for the experiments. Thank you very much. I'm happy to take any questions.
Thank you, Ruben, for your presentation. As anticipated, it was an excellent presentation with a lot of data. The work you are doing in your lab is impressive to trying to understand which is the role of all these pathways and signaling and metabolic pathways in the progression of liver diseases. So thank you so much for being here today with us and sharing all those data. Now we have time to questions and answer. I have seen that Lisa has been responding to the question that were on the chat directly, but there is still some questions that were not answered. There were a lot of clinical questions regarding how to implement all the criteria and all the aspects that Lisa was explaining through the presentation, but there are still some questions that were not addressed there. So we have time now, if you want, Lisa, it's your opportunity also to respond all this some questions that were not responded yet. So there is one saying, thank you for your talk. In patients with type 2 diabetes and or class 2 or 3 obesity, how do you suggest to screen for muscle D?
Yeah, so in obesity, of course, the ultrasound-based methods will be limited, as I mentioned. So I think maybe Lina can assist me also, but I think maybe MRI-based technique is preference, but also the FIB4 is not the first line. I see it as a surveillance because in some patients, steatohepatitis may be present also in normal transaminases.
Yeah, thank you, Lisa. If you allow me, the problem is that when we have biochemical findings of elevated transaminases, the problem is already. Yeah, the problem is there. I think that we should ask for a liver ultrasound for all patients with obesity. Of course, we should ask for lab tests for liver tests. And in my opinion, FIB4 is a very good index, easy to get, but still we need more non-invasive markers for good screening of patients with this condition. But a liver ultrasound is very important because we also need to see if there are any other problems. We give GLP-1 receptor agonists to these patients. We need to know if there are problems that predispose to pancreatitis. We need an ultrasound for many reasons. So I think that and MRI is good, but it's very expensive and cannot be implemented in all patients, of course.
Yeah, and for other biochemical markers, I think that also importantly we have to look on each patient individually. So if the triglycerides are rising, and so it's also showing us, and so in complex to see the patient, not just FIB4. Yeah, it is in the guidelines, very underlined, but yeah.
Yeah,
So,
now you are mentioning the transaminases, that there is also another question regarding that, is saying, is AST-ALT ratio useful? And what about gamma GT?
yeah, SLT-LAT ratio can help us to get some indication of the alcohol consumption. Gamma GGT, yeah, it is also a liver marker, and depending on the source. So of course, if these markers are changed, it raises also our suspicion that we should screen the patient and investigate and do the surveillance. Lina, do you have?
Yes, I agree.
Yeah,
But if the first marker that is increased in people with this condition is ALT. AST is mainly increased in people who have liver disease due to alcohol high consumption.
Yeah.
This is something that we teach to students, and we need to keep it simple. ALT is liver. You know, it's from fat. I mean.
Okay, so we can be jumping from clinical to translational because there are some questions from Ruben. There is a question saying, is there different expression of promoter genes in ethnicities with increased risk of muscle D, for example, in South Asians?
It's a good question. I know that there are some mutations that are more prevalent in different ethnias, but I do not know if the expression of genes is really different. I'm not aware of studies on that, but maybe there are some. Honestly, I don't know. I know about mutations, but expression, I'm not sure.
Yeah, I think we still need to learn a lot about this disease because it's a disease that can appear in different backgrounds, from normal weight people, in obese people, in people with diabetes, I think we need to investigate a lot about which is the origin of the disease and how it progressed. So there are still a lot of open questions. So we have more questions, probably for Lisa. Any advantage of fibrates in muscle D, or have they been excluded as an effective agent?
Have not been excluded. There are studies showing some benefit, but no very big studies.
There is another question probably for both things. For both the clinical and research-based talks, what about the role of low carbohydrates diet in muscle D?
Low carbohydrates, I don't know. I don't know. I'm not aware of studies on that, neither. I'm not an expert or neither in diet. Most of the studies focus in fat and sugars. That carbohydrates, low carbohydrates, more specifically, I don't know if there is a link with muscle D. Honestly, I'm sorry, but I don't know.
You know, carbohydrates are directly converted into glucose, so it's something that we recommend in the clinical practice, especially in patients with type 2 diabetes. But yeah, we need more evidence on that. But we need to do something, and it's one of our clinical advice, a clinical advice that we usually give to reduce a little bit the percentage of carbohydrates during the everyday diet.
Okay. One more question. What's your expectation, I think, Lisa, for the definitive management of oral and injectable GLP-1 analogs in muscle D?
Well, so as I already mentioned in the presentation, so of course we expect effect because weight loss appears happens in quite many patients, and also the pathogenetic mechanism of GLP-1 receptor agonists is very beneficial for muscle D. But as outlined, we are awaiting the results of phase 3 trials. So phase 2 trials in many cases are promising. And also for dual agonists, tirzepatide, for instance.
I think that there are a lot of promising clinical trials now going on, right?
Yeah.
So, Ruben, a question from Dr. Nogueiras. Do you know if any microbial metabolites affect the expression of P63?
We didn't check, but I'm sure that those metabolites that reach the liver will affect the expression of P63, because P63 is a transcription factor, but it is also a gene responsible for stress. So when the cell is somehow stressed, the levels of P63 are increased. So this means that whatever happens to the cell, the levels of P53 and P63 are likely changed. And those metabolites, independently of what they will do, because there were some metabolites doing beneficial effects or doing detrimental actions, I'm pretty sure that P63 and P53 will respond to them.
Okay. A really short question, I think, with a quick answer. Is liver fibrosis reversible?
Only initial stages.
Only. Okay. And one really general question. What about the discovery of new drugs for muscle D? I think a really open question that a lot of scientists and clinicians are trying to respond, right?
Yeah, well, I in that case, if I may say my opinion, I think that in addition to these incretins, we now have resmetirom, as Elisabetta has shown in the presentation, which has been the first one that has been approved by the FDA. And there are many other clinical trials, phase 1, phase 2, that I will assume that at least some of them will go to further stages. And well, I think that there will be more drugs definitely in the market within the next years. There are so many companies and teams working on that, so I'm sure that there will be more drugs.
Yeah. We will need more, right? Because even if we have resmetirom to be useful, it is only useful in 30% of the patients, right? In the clinical trial. So obviously there is still a huge hole to be covered by other drugs. So let's see what happens.
Yeah, and the reversion of the fibrosis is just one stage.
Yeah.
So it's good, but it has some limitations.
Exactly. Yeah. So, Lisa, one question. Do you have any case treatment of resmetirom?
In Latvia, it's not yet available. Not yet available in our country.
Okay. Ruben, how much does change in microbiome affect epigenetics in animal models? Do you have any clue about that?
Microbiome and epigenetics. Oh, that's a nice mixture. I would say that epigenetics can affect gut microbiota. Whether gut microbiota can affect epigenetics, well, sorry, thinking about it again. Yes, gut microbiota for sure is affecting the expression of microRNAs, and microRNAs are related with epigenetics. So yes, there is a connection there, and there are studies showing that gut microbiota can affect microRNAs and epigenetics. Yes.
Okay, one more question. I think it's a quick question. Is muscle D sometimes possible even in patients with BMI in the normal range?
Yes, it
Obviously.
is sometimes possible.
Probably just the last question, just to try to keep along on time, is if ALT and AST increases up to 3, Norma, it shows that fibrosis get worse.
It shows us that the liver damage increases. So for fibrosis, it is kind of LAT and SLT alone cannot give us precise information on fibrosis stage.
Okay, so as you can see, there are a lot of questions. This is a topic that is generating a lot of doubts and questions from the audience, but I think that it's being 7:30, 8:30 in Latvia, right, Lisa? I think it's time to end this session. So I would like to thank again both speakers for this really interesting spotlight on science. Thank you for sharing your knowledge and your research with us. Lina, do you want to say something to?
Of course, I think that it was a very interesting session, too many questions, excellent lectures. I would like to thank the speakers and all the participants. I think, Manuel, that this is the last spotlight for 2024. So
Exactly.
it's early, but we will wish everybody Merry Christmas and happy festive season and we will meet again with the new year, during the new year.
Okay, so yeah, absolutely. This is the last one. We will come back in 2025 with new spotlights on science. We will be announced really shortly. The first one will be in February, 27th of February, but you will know about it pretty soon. So thank you so much to both the speaker, to Lisa, Lina, to all the ESE members and all the assistants for being here. It was, I think, a great year of spotlights, and as Lina told you, happy end year and happy new year. Thank you so much for being here. Bye bye.
Bye.
Thank you. Bye bye.